Welcome everyone. We are going to lecture 16 of Introduction to Food Chemistry, and in this lecture we want to understand more about protein chemistry and specific concepts that are involved in protein food chemistry. We have noted earlier that proteins are essential for muscle and tissue formation, for enzymatic activity, and also for metabolic regulation. And we again recall that proteins are composed of 20 amino acids arranged in chains. Above is the structure of an amino acid, and we noted that it has the carbon, which is connected to a carboxyl N, to a nitrogen, or the amine end, and then we have the hydrogen side, and then the other group that determines the specific properties of this particular amino acid. The major sources of proteins, still we noted earlier, we have the dairy products, meat, we have eggs, those are the animal sources, and then we also have the plant sources that include the legumes, the cereals, the oil seeds and nuts. Proteins in food are very vital for the structure, the function, and the nutrition, and their behavior is governed by amino acid composition and structure. The charge and pH dependence. We have the isoelectric point, whereby you find that the net charge is 0. This is a pH point where the net charge is 0. And then solubility is lowest at the isoelectric point due to the minimal ionization. pH changes alter the structure due to electrostatic repulsion. Hydrophobicity and hydrophilicity. We noted that we have the hydrophobic and the hydrophilic amino acids, and the proteins are self-organized. You find that the polar side chains are facing outwards and the non-polar side chains are inwards. The example here we have is casein, which forms micelle-like structures. Casein is a milk protein. Denaturation of proteins can be caused by heat, by pH, salts, heavy metals, detergents, and alcohols. And denaturation causes reduction in solubility, increase in viscosity, and reduction in water binding capacity, and also causes the proteins to lose their activity. Maillard's reaction is one of the important reactions in food processing involving proteins, and we said it occurs between amino groups and reducing sugars. And this reaction is accelerated in alkaline conditions and in low water content. It is responsible for enzymatic browning, whereby you see the bread crust is brown, and then also roasted meat. High lysine or histidine equals to stronger Maillard effect. Hydration and solubility. This can be determined by the protein, water versus the protein-protein interactions. There is a higher charge density and lower hydrophobicity, which leads to better solubility of proteins. And water is essential for protein flexibility and function. For proteins to be stable under heat conditions, it is a very important quality for processed foods, because we note that heat causes protein denaturation, and during denaturation there is unfolding of the protein structure, leading to aggregation and eventually thickening or gelling. This is very practical in eggs. When you are frying eggs or when you're boiling eggs, you can see how the structure suddenly becomes compact. From a liquid, it solidifies. Then this stability can be improved by hydrolysis or mineral chelators. The viscosity of protein solutions. Viscosity refers to resistance to flow, and it affects the texture and the processing of proteins. And this is related to the molecular mass of this protein, the hydration status, and also the protein-to-protein or the protein-to-solvent interactions. The viscosity can be affected by pH, concentration, the temperature, and also the ionic strength. Gelation basically refers to a transition from fluid to a 3D viscoelastic gel, and it can be induced by heating that leads to protein unfolding and eventually aggregation. It can be influenced by pH, protein type or concentration, calcium, and the Ara-SH groups. And this is a key process that is used for processing products such as yogurt, cheese, tofu, and eggs. Emulsification and forming. Proteins stabilize interfaces, for example, oil-water or air-water interface, and they are good at unfolding and occupying interfacial zones. The example we have here is marriage. This is an egg white that traps air and water during whipping. And finally, we have what we call flavor binding. Proteins bind diverse flavor molecules due to the variety of their side chain interactions, the polar, the nonpolar, and also the ionic. So these are the few concepts that are involved in the processing of proteins involving food chemistry. Thank you.